we have this incredibly revolutionary targeted drug for acute myeloid leukemia, and it works brilliantly. I mean, until the cancer literally shapeshifts its entire biological identity to just, become completely invisible to the medication.
Yeah, it really forces us to completely rethink what drug resistance actually looks like. Because we're no longer just looking at a static target that mutates its DNA to block a drug. We are looking at a really incredibly fluid, adaptable system.
Right. So welcome to the deep dive, everyone. Today, we are dissecting a massive, really paradigm-shifting study that was published in Cell Stem Cell. It's by Wacławicz and this extensive team of researchers. And what they did was they analyzed transcriptomic data from over 150 patients with acute myeloid leukemia, or AML. And our mission today for you, the listener, is to understand exactly how these cancer cells are pulling off this biological espionage, and more importantly, how scientists are mapping these secret identities to basically corner the disease.
Exactly. And to understand the escape, we really first have to map out the exact mechanism of the trap we're setting, right? So for older or maybe physically frail patients diagnosed with AML, aggressive induction chemotherapy just isn't an option.
Right, their bodies just can't handle it.
Exactly. So the standard of care has shifted to this targeted therapy, specifically a drug called Venetoclax, and that's usually combined with a hypomethylating agent like azacitidine.
And mechanically, if you look at the biochemistry, this combination makes perfect sense. The azacitidine, it acts by removing methyl groups from the cancer's DNA, so it's essentially stripping away that epigenetic silencing and forcing the cancer to express genes that make it more vulnerable.
Yeah, that's exactly right.
And then Venetoclax comes in as this precision strike. It's what's called a BH3 mimetic. So it specifically targets and inhibits this protein called BCL2. And BCL2 is this crucial anti-apoptotic protein, basically an anti-death protein, that these leukemia cells overexpress just to keep themselves immortal.
Right. But the clinical reality, it's significantly darker than the biochemical theory. Because while that Venetoclax and azacitidine combination, it shows really fantastic initial response rates, the durability of that response is a huge problem.
Yeah, the numbers are rough.
They are. Over 60% of these vulnerable patients actually succumb to the disease within 24 months. And even more alarming than that, roughly 35% of patients present with upfront resistance. So the drug combination just simply fails to trigger apoptosis or cell death from day one.
Which, that points directly to the leukemic stem cells, the LSCs. These are like the self-renewing drivers at the very top of the leukemia hierarchy. So I always think of it like this analogy. Traditional chemotherapy is like a lawnmower, right? You're cutting down the visible cancer cells. But the LSCs, they're the deep dandelion roots. If you don't kill the root, the weed just comes right back.
That's a really good way to visualize it.
But I've got to push back here. Because if Venetoclax is explicitly designed to be this ultimate root killer, to bind to BCL2 and force the LSC to die, why are so many of these dandelion roots surviving?
Well, that is the core driver of Wacławicz's study. The researchers hypothesized that the LSC population in a patient is not just this single monolithic entity that relies solely on BCL2.
Oh, so the roots are different.
Exactly. To prove this, they utilized single-cell RNA sequencing. So they captured the active transcriptome, like the real-time genetic instructions these cells are reading, in over 150 patient samples. And then they took that data and overlaid it against a map of normal, healthy human blood development.
Right, so looking at the data, it seems they uncovered that the leukemia hierarchy isn't just one type of rogue cell. It's actually fractured into 4 distinct branches that perfectly mimic these specific stages of healthy blood creation. So let's unpack this for the listener.
Sure.
3 of these subtypes are what the researchers call classical LSCs, right? And they all share the specific surface marker, a flag on their surface called GPR56.
Right. And the transcriptomic signatures of these 3 classical states, they are sort of frozen in different stages of normal development.
Okay.
So first, you have the HSC LSCs, which are transcriptionally locked in the earliest, most immature hematopoietic stem cell phase. Think of them as the raw recruits. Then second, the LMPP LSCs. These are primed down a pathway that would normally create lymphoid or myeloid cells, so kind of like the standard infantry.
Got it. And the third?
The third are the MEP LSCs, and these are halted in this specific progenitor state that usually goes on to produce megakaryocytes and red blood cells. You call them the Navy.
Okay, so that gives us our 3 classical branches, Army, Infantry, Navy, basically. But the data shows a 4th, highly divergent branch, the non-classical mode LSCs, the special ops.
Yes, the special ops. And the acronym stands for mature, monocytic, and dendritic-like. This subtype fundamentally breaks the rules. Well, these cells completely lack that GPR56 marker found on the other three branches. Instead, they express a distinctly different surface marker called LMP5. Transcriptionally, they resemble much more mature white blood cells, but they still retain that vicious self-renewing capacity of a primordial stem cell.
So if you're a clinician listening to this, you aren't just treating quote-unquote AML. You are treating a disease driven by one of four entirely distinct cellular states. But, let's do a quick recap here. We've mapped the four branches. We know the roots are different. But simply mapping these states doesn't explain the resistance. Like, what is actually happening at the molecular level that makes an MEP cell inherently bulletproof to Venetoclax, while an LMPP cell just shatters upon contact?
That brings us to a really beautiful piece of biochemical modeling introduced in this paper, which is called the MA score.
The MAC score.
Right. The researchers recognize that apoptosis, programmed cell death at the mitochondrial membrane, is basically a thermodynamic tug-of-war.
Okay, a tug-of-war.
Yeah, so Venetoclax neutralizes BCL2. But BCL2 has siblings in the anti-apoptotic family, specifically proteins called MCL1 and BCL-xL. And Venetoclax cannot bind to MCL1 or BCL-xL. Its chemical structure is just too rigidly specific to that BCL2 pocket.
Okay, so if you're a cancer cell under attack by a drug that destroys your BCL2 shield, what do? You grab a different shield.
Precisely. And so the MI score is essentially a mathematical ratio of vulnerability. It calculates the transcriptomic expression of the drug's target, BCL2, and then divides it against the expression of those alternative resistance shields, the MCL1 and BCL-xL.
Right, so when they applied this MES score to the four distinct LSC identities, the resistance mechanisms just lit up immediately.
They really did. The LMPP LSCs, those infantry cells stuck in that early lymphoid prime state, they naturally produce an overwhelming amount of BCL2 relative to the other proteins.
Yeah.
So they have an extremely high MSE score.
Right, because their entire developmental state, like the stage they are frozen in, dictates that BCL2 is their primary survival mechanism. So if you have a patient whose leukemia is primarily driven by those LMPP cells, Venetoclax is going to look like a miracle drug, right? Yeah. It perfectly neutralizes their only shield.
Exactly. But then look at the other branches, the MEP LSCs, the ones on that megakaryocyte and erythroid track. Their biological state dictates a natural heavy reliance on BCL-xL.
Okay.
And the mode LSCs, that rare monocytic special ops group, they rely almost exclusively on MCL1. So you introduce Venetoclax to a microenvironment dominated by MEP or mode cells, you're essentially attacking a reinforced concrete bunker with a glass hammer.
Wow.
The drug is working perfectly, but on a protein the cell isn't even using to survive.
Which perfectly explains that 35% upfront resistance rate we talked about at the start. Those patients had disease driven by MEP or mode cells from literally day one. But, okay, here's the biological mystery that this paper completely dismantles. What about the 60% who respond beautifully to Venetoclax at first, but then relapse super aggressively a year later? Like, if their disease was driven by those vulnerable LMPP cells, why are they coming back?
So to answer that, the researchers analyzed longitudinal data. They performed that single-cell RNA sequencing on samples from the exact same patients at their initial diagnosis and then again at the point of relapse.
Right.
And they documented a mechanism that represents a massive paradigm shift in oncology. It's called lineage plasticity.
Okay, I want to emphasize how wild this is for the listener, because when we normally think of cancer acquiring drug resistance, we usually think of like Darwinian selection acting on genetic mutations, right? Like a random DNA error alters the shape of the BCL2 binding pocket, making the drug bounce off, and then that one mutated clone takes over the whole bone marrow.
Right, that's the classic model.
But that is not what's happening here. The DNA sequence itself isn't actually changing to cause this resistance.
No, the genome remains relatively stable in this context. What changes is the epigenome and that active transcriptome. Under the immense selective pressure of Venetoclax, which is, aggressively clearing out all those BCL2-dependent cells, the surviving LMPP LSCs actively rewire their transcriptional programming. They undergo what the researchers call an MEP shift.
Okay, so think of normal blood development like a massive rail yard. You have a master switch at the top, and trains are routed down different tracks to become different cell types. Venetoclax basically parks a massive blockade directly on the LMPP track. But because these are stem cells, they retain epigenetic fluidity. They actively throw a biological switch, reverse out of the LMPP state, and route themselves down the MEP track. They literally change their cellular identity, like changing clothes, to bypass the blockade.
It's incredible. And because we know that the MEP state is inherently dependent on BCL-xL instead of BCL2, the act of shapeshifting naturally swaps their survival shield.
Oh, wow.
Yeah, so by the time the patient relapses, the drug is still in their system, perfectly inhibiting whatever BCL2 is left. But the cancer no longer cares. It has entirely transitioned into an MEP state and is comfortably surviving on BCL-xL.
That is just terrifyingly smart. But the researchers didn't just document this rail yard maneuver, right? They figured out how to derail it. They utilized patient-derived xenograft models. So basically taking human leukemia cells and engrafting them into mice to test combination therapies in vivo.
Right, and the hypothesis there was pretty straightforward. If the cancer's main escape route is shifting to a BCL-xL dependency, we'll block that exit before they can use it. So they treated these engrafted models with Venetoclax combined with an investigational BCL-xL inhibitor from the very start of the experiment.
So you bomb the LMPP track and you blow up the MEP track simultaneously.
Exactly, and the results were profound. This dual blockade completely prevented the lineage plasticity from occurring.
Wait,
Completely. The leukemic burden in the bone marrow of these mice was reduced by up to 10-fold compared to treatment with Venetoclax alone. And honestly, even more impressively, this upfront dual inhibition was highly effective in models bearing TP53 mutations.
And TP53 mutations are notoriously bad, right?
Oh, absolutely. They traditionally represent the most aggressive, therapy-resistant forms of the disease. But by anticipating the shapeshift, they trapped the cancer.
Okay, let's do another quick checkpoint for everyone listening. We've mapped the classical cells. We structurally handled the MEP escape route, the shapeshifters. But I'm kind of stuck on this fourth group, the non-classical mode LSCs, the special ops.
Yes.
Because if they don't even use the GPR56 marker and they rely entirely on MCL1, they're essentially playing by an entirely different rulebook. If they are so inherently refractory to our most modern targeted therapies, why weren't they flagged as a massive biological threat like decades ago?
That's a great question. And it's because the older standard of care, those intensive cytarabine and anthracycline induction regimens, they were blunt instruments. They caused widespread cytotoxic DNA damage. The mode cells responded averagely to that broad-spectrum toxicity, so they never really stood out in the survival curves.
I see.
It is only now, when we transition to highly specific, targeted BH3 mimetics like Venetoclax, that the mode cells are violently exposed. They just simply ignore the targeted therapy.
Right, and they make up about 5 to 10% of all AML cases, which means, thousands of patients are currently being given a targeted therapy that has zero biochemical chance of working.
Yeah. And this brings us to perhaps the most immediate, highly translatable clinical victory in the entire study. Because look, you cannot run a complex, multi-day, single-cell RNA sequencing protocol on every single patient who walks into an oncology ward just to find out if they have mode cells. The turnaround time is simply too slow for an aggressive leukemia.
But the researchers found a shortcut.
They did.
We mentioned earlier that the mode cells express a unique surface marker called LMP5.
Exactly. LMP5 acts as a highly visible diagnostic flag, and its presence can be rapidly detected using flow cytometry.
And that's pretty common, right?
Very common. Flow cytometry is a ubiquitous technology. Pretty much every major diagnostic laboratory has one. You take the patient's bone marrow aspirate, tag the cells with a fluorescent antibody specific to LMP5, run it through the cytometer, and within hours, you have a definitive answer.
Wow, within hours?
Yeah. If that specific cell population lights up, the clinician instantly knows they are dealing with a mode-driven leukemia.
So, okay, put yourself in the clinician's shoes. You see the LMP5 flag.
Yeah.
You know Venetoclax is useless because those mode cells are hiding behind a massive MCL1 shield. What is the clinical countermove? Like, how do you actually strip an MCL1 shield?
Well, the researchers conducted a vast screen of cellular signaling pathways to find a vulnerability. And they discovered that mode LSCs are uniquely addicted to the MAPKIR signaling pathway.
Okay, that's a mouthful.
Yeah, specifically the kinesis MEK1 and MEK2. This pathway is continuously hyperactive in these cells. And crucially, this MEK signaling is exactly what biochemically stabilizes their MCL1 protein, preventing it from just degrading naturally.
Okay, I follow. So if you introduce a MEK inhibitor like a binimetinib, which is already used in other cancers, right?
Yes.
You sever that signaling pathway. The MEK inhibitor doesn't necessarily trigger cell death on its own, but it destabilizes the MCL1. It physically strips the shield away from the mode cell.
It completely disarms them. In their in vivo models of mode MOL, Treating with venetoclax and azacitidine alone failed completely.
Right.
Treating with the MEK inhibitor alone also failed to induce a deep response.
Right.
But when they combined all three, venetoclax, azacitidine, and the MEK inhibitor, the synergistic effect forced the cancer into profound remission.
Right.
They basically chemically stripped the MCL1 shield, resensitizing the cell and allowing the venetoclax to execute that apoptosis program.
Which fundamentally changes the way we need to think about risk stratification in the clinic. I mean, right now, the dominant paradigm for predicting how an AML patient will fare relies heavily on bulk genetic profiling, right? We look for specific structural mutations in the DNA. Like if the pathology report shows a mutation in the NPM1 gene, that is clinically categorized as favorable. The patient is generally given a good prognosis. Conversely, a TP53 mutation or a highly complex tangled karyotype that is universally recognized as high-risk adverse genetics.
But what this study conclusively proves is that analyzing the genetic sequence alone is only looking at half the battlefield.
Right.
The genetic mutation tells you the blueprint of the cancer, but the LSC state, the specific transcriptomic identity we've been discussing, that dictates exactly what the cancer is doing in real time and how it will interface with the drug.
Exactly. Genetics is what it is. State is what it's doing. So you can have a patient with that favorable NPM1 mutation. Historically, a doctor would assure them that venetoclax has a really high probability of success. But if that patient's transcriptomic profiling reveals their leukemia is locked into their resistant MEP-LSC state, heavily reliant on BCL-xL, that patient is going to fail therapy rapidly. Their state completely overrides their favorable genetics.
And, the inverse is equally true. A patient with notoriously high-risk genetics might harbor leukemia that is strictly in the highly vulnerable LMPP state, meaning they could experience a deep, prolonged remission on standard venetoclax therapy. And to demonstrate this, the researchers built this unified dual-layer risk stratification model. They synthesized the traditional genetic mutation data with this new LSC state data.
And the divergence in the survival curves when they did that is just absolutely staggering. When you look at patients in the newly defined combined favorable group, meaning they possessed good genetics like NPM1, alongside those highly vulnerable LMPP LSCs, their median event-free survival extended to over 1,100 days. That is over 3 years of holding an aggressive leukemia at bay.
It's incredible. But then when you look at the combined adverse group, those harboring the LMP5 positive mode cells, or patients with high-risk genetics combined with the biologically resistant MEP or HSC states, their median event-free survival plummeted to just 89 days.
89 days. I mean, that isn't just a statistical difference. That is a brutal biological reality check. It proves that administering venetoclax to that adverse group without targeted adjuncts is just a completely futile effort.
Which is exactly why oncology is racing toward biomarker-guided personalized medicine. The days of standard trial and error regimens, they're ending.
Right.
By utilizing flow cytometry and transcriptomics at the time of diagnosis, we can accurately calculate the MA score before a single drop of medication is administered.
Right.
We can anticipate the exact thermodynamic shield the cancer is using and preemptively administer the precise inhibitor, whether that's a BCL-xL inhibitor to block plasticity or a MEK inhibitor to strip away MCL1.
We are finally learning to see the board exactly how the cancer sees it. We're predicting the shapeshift before it even happens. But as we synthesize all of this, I can't help but be struck by the sheer, almost terrifying ingenuity of this disease. It isn't just a chaotic mass of rapidly dividing cells.
No, not at all.
It is highly organized. It intentionally hijacks the developmental plasticity of normal human biology. It remembers how to act like a primordial stem cell sliding across those epigenetic tracks, dropping one shield and picking up another, purely to outmaneuver our most advanced synthetic drugs.
It is a really profound reflection of our own fundamental biology. The cancer is utilizing the exact same epigenetic mechanisms that build a complex human body from a single embryonic cell, but it's weaponizing that flexibility to dismantle the host.
Which leaves me with a final provocative thought for you to carry forward, our listener. If leukemia relies on this incredible lineage plasticity to survive the selective pressure of our most toxic targeted drugs, what happens when we completely invert our therapeutic strategy? Like, what if, in the future, the goal of an oncology ward isn't to design a better poison to shatter the cancer cell? What if we design drugs that actively force the cancer to complete its differentiation pathway? If we can manipulate these epigenetic tracks, perhaps the ultimate cure isn't killing the cell, but acting as a master biological director. forcing the deadly stem cell to shapeshift into a harmless, terminally differentiated mature cell that naturally ages and dies.
You are describing the absolute frontier of differentiation therapy. And with the transcriptomic maps provided by Wachluchek and this remarkable team, we now actually have the exact coordinates to start pushing those biological switches.
It is a massive step forward in understanding the enemy. Thank you for joining us on this deep dive into the hidden identities of leukemia. Keep questioning the world around you, keep looking for the mechanisms beneath the surface, and we will catch you next time.